Chapter 6, Part 2, Where do prokaryotes grow? We've already described some terms that we use for oxygen requirements. Let's see a few more examples and then some additional terms that we use to describe prokaryotic growth. Here we have a couple of examples of modified terms for oxygen requirements. We've already defined microaerophilic, meaning just a little oxygen. There's an additional term, capnophilic, meaning carbon dioxide loving. There are some organisms, particularly bacteria that cause infections, or what we would call pathogens, that do require or prefer a 5% carbon dioxide atmosphere compared to the 2 or 3% that's usually available. So in order to increase the CO2 level, we often put them in containers that we call candle jars or CO2 jars. A candle jar is one in which we will place our stack of petri dishes or cultures. We'll actually take either a gas pack that can increase their carbon dioxide level, or we can simply put in a candle that we've lit with a flame, seal the container, allow the candle to burn or undergo combustion, which will consume some of the oxygen, and in the process of combustion, release a little extra CO2. Hence, candle jar. That's illustrated on the right-hand side, and we can also use the device that's illustrated on the left-hand side of the photo that can be used either as a candle jar or CO2 jar for a capnophilic or oxygen lower, CO2 higher environment, or we can even use the device, either one of them, to create an anaerobic environment, or an anaerobe jar in which we could grow an obligate anaerobe. With either a gas pack or some other extra kind of device, with an anaerobe jar, we have to completely remove all of the oxygen for the obligate anaerobes to grow. And again, this is a sealed container in a specialized structure for the growth of those obligate anaerobes that need that special oxygen-free environment. Now here we have 3 different organisms, Staphylococcus aureus, Clostridium butyricum, and Pseudomonas aeruginosa. They have been grown in 3 different oxygen-requiring conditions. The first column is the growth of all the organisms in an oxygen-saturated environment, typical atmospheric oxygen. The center column would be a candle jar where we have increased carbon dioxide to 5% of the atmosphere and a little bit less oxygen. And then we have on the far right column the plates placed into an oxygen-free environment, or an anaerobic gas pack was used to create an anaerobic oxygen-free environment. How would you describe the growth on these 3 plates? And then what kind of phrase or term would you use to describe the oxygen requirements for Staph aureus versus Clostridium versus Pseudomonas? Well, if we look at the growth of Staph aureus, we'll notice that Staph aureus can grow if oxygen is present. And on the far left column at the top, we can see that the colonies appear very dark or bright yellow, and we have very luxuriant growth. Same thing's true for growth in the candle jar with 5% carbon dioxide and a little bit less oxygen. Even in the gas pack, anaerobe jar, or anaerobic environment, we still have growth of Staph aureus. It's not nearly as luxuriant and not nearly as bright in color, but we still have growth. So this would definitely be an example of a facultative anaerobe, growing with or without oxygen. Clostridium butyricum, on the other hand, if you'll notice, does not grow at all, either in oxygen or even a little bit of oxygen in the candle jar. So this one is definitely an obligate anaerobe. In comparison, we look at Pseudomonas aeruginosa. This is an obligate aerobe. It definitely has to have oxygen present. Even in the candle jar, with a little bit less oxygen, it still grows. Notice that we have a slight difference in color or pigmentation with the oxygen-rich environment versus the slightly lower oxygen in the candle jar. Here we have some more tubes for comparison. Can you tell which tube is the obligate anaerobe? Which one is the obligate anaerobe? Is there an aerotolerant anaerobe in the mix of test tubes? Sure enough, the test tube on the far right shows the growth of an obligate anaerobe. There is no growth in the top of the broth. All of the growth is where there is no oxygen present. The aerotolerant anaerobe, an excellent illustration of that type of growth, is the test tube second from the right. Growth all the way from the top to the bottom, whether oxygen is present or not. In addition to terms describing oxygen requirements, we have other terms that describe temperature requirements and pH, or acidity and alkalinity. When describing terms referring to temperature requirements, we're going to use the same suffix, phile, meaning loving, and then add different prefixes to that root word or that suffix to tell us what kind of temperature. A psychrophile is a cold-loving microorganism. A mesophile loves sort of middle of the range temperatures, like body temperature. And the thermophiles, or the extreme hyperthermophiles, like heat. So a thermophile is a heat lover, and a hyperthermophile is an extremely heat-loving organism. If we graph out the growth, temperature versus rate of growth, we're going to find that the psychrophiles prefer cold temperatures, temperatures we'd even find in the refrigerator, anywhere from -4 to 30 degrees Celsius. The mesophiles prefer body temperature, ranging from about, let's say 10 to 15 degrees to about 45 degrees Celsius. And the thermophiles, anywhere from 40 to 50 degrees Celsius to 60 or 80 degrees Celsius. And the hyperthermophiles will grow easily at and above 80 degrees Celsius to 110 degrees Celsius. Now notice that the temperature range on the graph doesn't exactly match sometimes the temperature range on the text, or it'll vary from textbook to textbook. But needless to say, in general, psychrophiles like cold, mesophiles grow at temperatures that are typically human body temperature, and the thermophile and the hyperthermophile grow at very high temperatures. In addition to terms referring to oxygen or temperature, we also have two terms that we use to describe whether or not a microorganism prefers an acid environment, an alkaline environment, and that's acidophile if we like acid environments below a pH of 7, or alkaline environments with a pH above 7. Now, pH, acidity versus alkalinity, we'll have more discussions on what pH means in more detail in Unit 2. But needless to say, if you've ever worked with vinegar in salad dressing, vinegar is an example of a weak organic acid, or a very common type of acid that we use in our everyday lives. If you've ever used bleach or ammonia for cleaning, those are examples of alkaline materials. And yes, indeed, there are some microorganisms, as illustrated in the photos, that can grow in very extreme acid and alkaline environments. Here are two scientific names of microorganisms that include in their name what kind of growth environment they prefer. Thermoplasma acidophilum, thermoplasma referring to heat-loving, acidophilum, acid. So this prefers a hot, acid environment for growth. Alkaligenes faecalis, alkaligenes, alkaline, prefers an alkaline pH environment. Now the species name faecalis, got an idea where that might be derived? Well, feces. So fecal or feces, alkaligenes faecalis, we might expect to find in feces.